All right, so we'll go ahead and get started. Thanks very much for coming, everybody. My name is Megan Peters, and I'm on the program committee for CCN this year. And we're really excited to bring you all a new format for these keynote and tutorials. So I thought before we started, we should do just a very brief introduction of what this is and what we are doing at CCN this year. So CCN is all virtual, as you all have figured out, and consists of 3 different types of events, which are the Algonauts winners competition announcement, which happened last week, but if you missed it, you can go and have a look at the recording. And then we also have the generative adversarial collaborations, the kickoff workshops, and also a wrap-up or progress report workshop that's coming up this Thursday, where kind of 2 opposing sides get together and fight it out over how to resolve interesting controversies in the field. But today we're going to have our 3rd type of event, which is the keynote and tutorial. So this is like a keynote on steroids, and where you get to see all the cool stuff that's going on, and then you get to actually get your hands dirty and understand exactly how to make some of the pretty pictures that you get to see in the keynote. So I think I will just probably go on here. So the idea here is that we want to not only in these keynotes see the science and how it comes about, but we want to actually look under the hood of the methods and really be able to ideally apply those methods to our own science in the future. And so this will happen in a hands-on tutorial that will follow today's keynote, where you can actually open Google Colab, and there will be links provided in the chat at that time and on the screen. So you can open the Google Colab and actually run the code, and then maybe copy bits of it and understand how it works. And the idea here is not only to replicate the existing analyses, but also to be able to apply these analyses to your own research and to enable new approaches and new techniques that you might find exciting. And so there are a lot of benefits that we see here for the CCN community at large, that they can highlight all of the stuff that went on under the hood that led to the beautiful figures and graphs and results that we see in these brilliant papers that we'll hear about today. And so it highlights all of the choices and assumptions and interpretations and questions that were asked when those analyses were taking place. We also hope this will drive community engagement. The keynote and tutorials were driven by community submissions, where we solicited applications to give a keynote and tutorial, and then we selected the ones that would best cover the CCN community's interests, and also provide a really hands-on and tight tutorial experience that would be easy for a lot of different people to follow. And so we hope to be continuing to do this in the future, ideally going back to in-person stuff and not just virtual stuff, but we're hoping to continue to do this in the future. So next year, when hopefully we go back to an in-person conference, we will have this call again, we hope. And so we encourage people that if you like this, that you should apply and give a keynote and tutorial to help the CCN community understand the science that you're doing and how you got, again, those pretty pictures in your papers. And so this is the first of our keynote tutorials this year. We have 3 coming to you. The second one is tomorrow, a little bit earlier in the morning. And then there's a 3rd one that's happening later on. So please check the CCN website, ccneuro.org, and you'll find, just as you did today, the abstracts and also eventually the Zoom links so that you can join these keynote and tutorials at these times. And so we hope that you guys will have fun today, that you will actually get your hands dirty with the code, that you will run the Google Colab, and I think that's all that I need to say. So I'm going to stop there. And I'm going to introduce our keynote speakers of the day. So our keynote speakers are Fatma, Deniz, and Jack Gallant, and they are going to be telling us about some really exciting work on Voxelwise modeling. And then that will be followed by a lengthy hands-on tutorial where you can really get your hands dirty. So That's it. Thank you all for being here, and thank you especially to the creators of the Keynote tutorial. We're really excited to see what you guys have to share with us.
Oh, thank you very much, Megan. That was a really nice introduction, and I'm also very excited about this tutorial format. I'm really happy that you guys are supporting this. It's a lot of the methods we use in MRI are pretty complicated, and it can be difficult to figure out exactly what's going on under the hood. And it's not a good idea generally to trust software packages that we have no idea what they're doing. It's better to be able to do it ourselves. So I think these tutorials give people a great opportunity to really get their hands dirty and figure out what's going on with these methods. Today, I'll be just giving a very brief introduction to this tutorial. The tutorial was organized by 4 of my spectacular current and former postdocs, and they'll be doing most of the work. I just wanted to give you a bit of context for this approach, Voxelwise Modeling. It's an approach for building encoding models for either voxels in MRI or for single units in neurophysiology. As you all know, the brain's a pretty complicated place. In naturalistic kinds of situations, there's a lot going on. For example, if somebody's driving through a town, they have to think about where they're going. They have to negotiate through the traffic. They have to figure out what road they're on. They have to perhaps talk with somebody else in the car while they're driving. They might be listening to the radio. They have a lot of planning that's long-term, planning that's short-term, and they have to avoid getting in accidents. In general, it's a fairly complicated sensory motor decision-making task. So something like driving elicits brain activity in a lot of different cognitive subsystems involving sensation, motor action, planning, abstract decision-making, and so on. As you can see, when somebody's in a driving simulator doing this task, this is just a recent movie that I happened to pull up. The patterns of brain activity that are elicited in this task are fairly complicated. In this flat map, red regions indicate regions of more brain activity, blue regions indicate regions of less brain activity. And as you can see, as the driver is negotiating the environment, the patterns of brain activity change quite dramatically and dynamically in real time. And these different patterns of brain activity represent different functional networks being switched into and out of the task as people are engaged in this naturalistic task. And there's really no simple sort of low-dimensional simplified experimental version of this task that you could build that would elicit activity in all of these different brain subsystems. So what we've tried to do in the lab is to build a framework that allows you to build very explicit quantitative models at the highest resolution possible that will recover as much information from these naturalistic scenarios as possible, and then enable you to model that in such a way that you can predict activity under new situations that you didn't use to create the model. And in all of this work that's really been ongoing in the lab for the past 25 years, we tried to follow best practices in statistics and data science to ensure that the results that we get are valid, that we minimizing type 1 error, minimizing type 2 error, maximizing predictions, and maximizing generalization. So if we are building if we're going to build a cognitive neuroscience method, something that's very general that we can use for a lot of things, we have a lot of criteria that we're trying to satisfy. For example, we want to recover rich functional maps, obviously, in the task. We would like to use naturalistic tasks. We like to create explicit computational models that are as mathematical as they can be, given the feature space that we're trying to model. We'd like to obtain a complete functional parcellation of the brain. We'd like to identify functional networks. We'd like to relate each individual subject to the group, since only a small fraction of the total variance in brain activity reflects the group model. We'd like to predict new data that we haven't collected yet, and we'd like to generalize outside of our tested conditions. And Voxelwise modeling provides a way to do all of this. Now, Voxelwise modeling actually comes out of an older method that was developed originally in neurophysiology to analyze single unit data collected from the visual system in awake behaving animals. And these data were collected under naturalistic conditions, and therefore some method was needed that would allow us to create feature spaces and explicit computational models that would account for these data. And it worked quite well in neurophysiology. And the Voxelwise modeling approach that you'll hear about today is just a generalization of that to MRI. So Voxelwise modeling has a lot of advantages and a few disadvantages. It provides a clear method that allows you to test several competing models, even if their feature spaces are not commensurate. It makes all the assumptions about the model and the model framework and hemodynamic coupling explicit. It permits the use of complicated natural stimuli and naturalistic tasks. It maximizes the mutual information between the experiment, which is, the subject and the task and the stimulus, and your computer. What it does is it maximizes the mutual information flow through this whole sort of experimental infrastructure per unit time money in order to maximize efficiency. It provides functional maps in each individual subject, which is going to be critically important as MRI moves forward and we move from just building group models to focusing more on individuals, especially for clinical applications. And provides measures of not only statistical significance, but also prediction accuracy and generalization. And just to compare Voxelwise modeling to other methods, it really satisfies a lot of problems, solves a lot of problems that other methods just can't approach. They don't even have any way to deal with that. And Voxelwise modeling can basically address all of these important issues in data analysis and modeling that whereas the other methods that are available can only address a fraction of them. So this method's been developed over about 25 years in my lab, and I've had a great crew of people that have been involved in this work. Most recently, 3 of my current and 1 of my former postdocs, Fatma, Deniz, Matteo, Visconti, Tom Dupre, and Mark Lescroart, have really worked very hard to create a set of software that anybody can use and a set of very, very good tutorials that can enable anyone to implement this method fairly easily. And that's great because there really hasn't been a software stack that's enabled people to do this easily in the past. So now for the rest of the talk, I'm going to turn it over to the crew, and Fatma will complete my keynote. Thank you very much for your time. Fatma?
Okay, can you hear me?
Yes, sounds great.
Okay, perfect. So I hope you can see my slides as well.
We can see your presenter view right now.
Presenter view. Okay, let me try again.
Yes.
Is this the slides now?
Yeah.
Okay, perfect.
Great.
Cool.
And you also shared, we'll check the audio for that one video.
Yeah, I did check that.
Great. Okay. All right. Thank you.
Okay, cool. Thanks everyone. Thanks Jack. Thanks Megan for the introductions. And yeah, my name is Fatma Deniz. As Jack mentioned, I'm at the TU building and a senior postdoc and PI at Jack's lab. And I thank everyone to be here today. I see like 50 participants. I think it's going to be a very cool session with a lot of learning about the doings of the Voxelwise modeling in the next few hours. Jack just gave a very nice introduction on the motivation behind the developments of Voxelwise modeling over the last few decades. And now I will just take the second part to describe some of the recent experiments that from the Gallant lab and also myself. And I try to show you that with some details in the methods that you will then see in the tutorials in more even detail by doing them as well. Okay, so before I start, I would like to draw your attention to the basically contrast between the more classical fMRI experiments and the Voxelwise modeling approach. In the classical fMRI experiments, we usually, or researchers in general, but we in general are interested in answering a particular question and a hypothesis that describes some cognitive functions, let's say. let's say researchers wants to know whether some parts of the brain is specialized in processing, I don't know, concrete versus abstract from the language processing example, then they would create a condition where they would show isolated words of abstract words like justice, belief, face, aspect, and another condition that is a contrasting condition with some other concrete words, for example, where such as apple, table, river, and house. And while doing that, brain activity is obviously collected using functional magnetic resonance imaging or any other imaging modality. MEG, EEG are possible as well, or ECOG. For electrophysiology. So we collect data under these conditions, and what we usually then do is we take average brain activity under one condition, average brain activity under the other condition, and then create a contrast map by basically subtracting these average activities and declare more or less that some of the, those brain regions that are showing up in this contrast map are highly activated for abstract conditions, abstract words, than for concrete words. So other variations, Jack mentioned them as well in his slide, exist for this also with the multivariate pattern analysis or RSA methods, which also usually use a few conditions that are collected under, under these that are collected using brain imaging modalities under these conditions, and looked at how these conditions are driving the brain activity in some portion of the brain. But in general, in the VM approach, we try to study cognitive functions using a more naturalistic approach. What do I mean by that? Well, the real world, when we listen to a talk, when we read a book, when we listen or talk to someone, everything is much more rich, right? The incoming information that we're processing is much richer, and we would like to know how the brain represents such complex and rich information, and not only one or two conditions, basically, that we design and test our experiments on. So we use neuroimaging to look into the brain while it's processing this complex information and develop computational models that can explain some of the brain data. For example, when you are a participant in one of our studies, we ask you to listen to a natural story, like a book, an audiobook, or read a book, or watch a movie. And while doing that, we are collecting brain activity. Here are just 3 example voxels time series that are that are as long as basically your story, for example, when you were reading a story during the scanning session. And then now, because we have this very rich brain imaging data that we collected when the subjects were reading or listening to a story, let's say, we can ask or start asking a lot of different questions. And these questions can be thought about like as the hypotheses that are usually used to design a classical fMRI experiment, for example, that I mentioned earlier. For example, if you are the researcher and from the language processing, maybe you are interested in semantic processing and how, or where semantic information is represented in the brain, you can create using different computational linguistics models, some semantic feature space or a syntactic feature space if you're interested in syntactic processing. In addition to that, you can use different types of information using, or like feature spaces using spatial temporal Gabor features for low-level visual information. So once we have a feature space that basically quantitatively describes our hypothesis or questions, then we are ready to map these features to the brain activity, which is basically the encoding part here, is a predictive model that explains brain imaging data given a set of features. And once we have such good encoding model, we can basically turn around that equation and use this model to create a decoding algorithm that can be even used to reconstruct from brain activity what picture or story the subject was reading, and to identify that. Okay, but the main focus today will be on the encoding modeling, and I will mention just briefly maybe the decoding part at the end of the talk. Before we start, here is an example short movie that Jack already showed a little bit. Part of it, this is only to orient you actually on the brain surface more or less. So we have a movie that was created using PyCortex, the main software that we're using to visualize our brain imaging data that was developed at the Gallant lab. And it will spin now one turn exactly. So here's the prefrontal cortex. Now it's the back of the part, and it will just, open up and create this flattened cortical surface. So to those who are not familiar with these images, the inner part of the brain will be the early visual cortex, or are in the early visual cortex. We have auditory cortex, for example, here with the temporal lobe lying here. Here's parietal lobe, and then prefrontal cortex is in the other part of the brain. And you will be seeing this flattened cortical surface throughout the presentation now today. Okay, so for the language processing, because I started also with the examples of the language processing, here are just some examples about some studies that use this more classical approach to fMRI in language research. For example, to study where different categories of words, as I mentioned, concrete versus abstract was a very prominent example. Binder and colleagues showed that some of these regions here in the temporal, sorry, I don't see my own screen now, temporal and parietal lobe are representing more, activated for concrete words than abstract words. Other examples are emotional versus neutral words. And more recently, also with the lab of Fedorenko, researchers have started to use more like sentence level or like sentence pre-representations in contrast to, for example, non-words or simple isolated words. I see this as an example, maybe in the to bridge the line to the vision community a little bit. In vision, we had like a time where we only were showing like objects, isolated objects that are totally out of context. Those are basically our single words in language. Whereas in the last decade, there has been a movement towards like using more at least naturalistic images, if not movies. And the sentences is going towards that direction as well in the classical approach. So again, in the VM approach, that should start later a little bit. Let's do this. So in the VM approach, we study semantic representations in the brain. If we study semantic representations in the brain, we don't restrict our experimental data to any specific conditions. So we can basically ask all these different questions about language processing. One of them is semantic representations when we collect a very rich data set. And this is an example of that rich data set that was collected by me and Alexandra Hood in 2016 and 2019, which is while subjects are basically listening and reading to the same set of English stories, monolingual subjects in this case, and you will see the presentation for the stimulus. In the listening condition, you will hear it, and the reading condition. And as Jack mentioned in the navigation experiment that he was showing earlier, the red regions are, or like those regions that are in red are more activated, whereas blue ones are less activated for this type of experiment.
Hit the brake at the red light. I flung open the door and I ran. I had no shoes on. I was crying. I had no wallet, but I was okay because I had my cigarettes.
Okay, so as you heard and read, it's a very natural story. It has a lot of, a lot of the typical things that we have in the everyday use of the language. It's not isolated words. Now they're all within context. These are usually 10-minute stories, and we have approximately 10, 11 stories for the whole experiment. So we collect a lot of data in each individual subject, and we always split our data to training, validation, and test data sets. Okay, so then once we have that collected data, we create a quantitative model that describe our stimulus. So that quantitative model in our case, because we're interested in semantic processing, for example, is describing or extracting for each stimuli word that we have in our experiment using some word embedding space features. A representation. So that word embedding space basically provides for each word some vector representation that is usually learned with some shallow or even deep neural networks. And the idea behind that is usually that words that appear in close proximity in natural language have just similar representations in this vector space, such as king and man here, for example, having very similar representations. So for each word in our stimulus, in listening or in reading conditions, we grab those words and get the semantic features basically for these different words that is basically approximating the semantics information and that we would like to map to the brain data. We also usually create, in addition to the main feature space, different additional feature spaces. So for this study, for example, we use syntactic feature spaces, phonemic, spectral, low-level visual, and letter descriptions of the words. We can always look into these individual models, and you will learn this in the tutorial part as well, and how much variance basically each of the model explains when you fit these feature spaces jointly in to the brain data. Okay, so once we have the feature space, we again try to map these features, the semantic and the additional feature spaces, to the brain responses using regularized linear regression. So we treat it as a regression problem that finds basically the best match, you can say, between the semantic features and the brain responses. And we do this for each individual voxel separately and for each individual subject also separately, which is a very unique property of Voxelwise modeling in this case. So then what we do is basically we estimate some semantic model weights in this case, or like estimate some model weights for listening and reading separately. And to then evaluate how well our model generalizes to new data, we just go back to the scanner and collect new data set. That is our held out test data set. That test data set has the same properties as your training data set, basically one other story that the just has not read or listened during the training session that we use for training the models. So once we collect this data, and there's a particularity for this data collected here, which is basically we use several repetitions of the same story, and everyone who have collected data in the audience will know that for each particular voxel, the data collected in one session and another session will not be the same. There's a lot of noise in the measurement and also in your brain. So basically, we try to restrict our test generalization performance only to those voxels that give you reliable response across several repetitions. So therefore, we collect always repetitions of our test story and then measure our generalization performance on those repetitions. Okay, so once we do that, basically now we have the estimated model weights for listening and reading separately, and then we can grab the same use the same semantic feature space, those word embedding spaces, to extract now the features, the semantic features for the test story that we were using, and just basically predict a time course per voxel of the brain responses that if we would use this model that would predict basically. We can do this separately for listening and reading. And remember, we went back to the scanner and collected the test data set, so we can actually really see how well our predictions were by finding a similarity metric between the predicted brain response per voxel and the recorded brain response for that test story. And this is our prediction performance or prediction accuracy for saying how well our model was in explaining brain activity. So this correlation coefficient, as the similarity metric here used, is then mapped on these like pretty pictures that Megan mentioned, but basically on the cortical surface for one individual person. This is one person's brain, one subject's brain, and for each individual voxel. And here, the brighter the color is, the better is the prediction accuracy, basically. And you can see there is like a temporal and a parietal and prefrontal cortex that are, in this case, representing semantic information because we are mapping the semantic model predictions onto the cortical surface. And another thing that you can observe on this picture is that we have the semantic information is very similar across listening and reading. And also we have very distributed, bilaterally distributed semantic information representation across the cerebral cortex. Again, this is one individual subject's brain in two different experiments where individual subject's variability is absolutely preserved. And we can also if we want, map our data to a template MNI space, which is a group average across subjects. And we see just the same pattern, but this is usually a post hoc analysis that we do afterwards, after we finalize our individual subject analysis. And if we want to draw quantitative conclusions for the group averages. Another idea that I would like to give you today is, here is an example of a meta-analysis using 120 different fMRI experiments that was combining these studies to understand where in the brain really semantic information is processed by Binder and colleagues in 2009. And you can see that this meta-analysis with all the different locations that one of like 120 different studies were showing high activity for semantic tasks are very distributed as well. However, if you would just go to one of these studies, you would only say, oh, okay, some portions of the prefrontal cortex is only highly activated, but you would not see this general pattern. Whereas when we do look at this naturalistic approach in combination Voxelwise modeling with one reading or listening, it doesn't matter in this case, as you saw, naturalistic experiment, we do find a similar network of brain regions for semantic representations. So this is also to illustrate the power of our methods here. And another beautiful thing that you can do, especially if you have two similar experiments that you are addressing similar questions, is basically you use the data and the models estimated in one modality. This is, for example, listening here, to make predictions about the other modality, which will, if this works, basically, it's a very good indication that when we listen and read a story, the semantic representations are independent of presentation modality. And once you predict the brain responses, again, the same thing applies here, that you are trying to find a similarity metric, or like using a similarity metric, you are comparing the predicted brain response to the recorded brain response. When we do that, we see that semantic representations are not only distributed across the cortex, but also, again, very similar and independent, very similar and are very independent of the modalities. So each voxel that is semantic selective is also modality invariant in this case. Importantly, I mentioned that many times, the analysis is done in single subject, as I said, and you can think about this, or like in general VM approach, as a replication of results across individuals and across different experiments as well. And here's just each individual subject doing this cross-model predictions that I just mentioned, with listening predicting reading or reading predicting listening. Okay, so these predictions are obviously very great, but we can even go further and examine what specific information each voxel is maybe representing. For example, for each voxel, we could examine the estimated model weights and study which features are basically preferred by that particular voxel, right? In theory, we can just go into the semantic model weights in this case and say, okay, this voxel likes some sort of like one particular concept. You can think about these semantic features if you want to, for example, what I mentioned earlier with the classical fMRI experiments, see where in the brain concrete versus abstract words are represented. You could just have a feature space that has like two columns here, and then very simply examine, at least for some portions of the voxels, which one this voxel prefers very directly, like concrete or abstract representations. However, it is very hard to inspect every single voxel, and in our case, the semantic features or our feature spaces are usually high dimensional. So we have, I don't know, around 60,000 to 70,000 voxels and some 300-400 or sometimes 1,000 features that also for every subject. So we have a lot of dimensions here, and to find some main axes across these different estimated semantic model weights, we can use dimensionality reduction techniques, such as one of them is principal component analysis, to reduce our dimensionality so that we can have like a semantic so-called principal component space that we then can use each individual's estimated model weights to map onto this semantic PC space and nicely visualized using different, for example, RGB color maps for the first 3 principal components, because those are explaining most of the variance in this case, and map those onto the cortical surface. So for each voxel, basically, we have a combination of these 3 different colors, which creates these semantic maps. And when we do that for these two different experiments, listening and reading, again, we see these very similar maps across experiments in individual subjects. You can see different colors are assigned to different concepts, and not only concepts are in some portions of the brain localized, such as the green, if you look at the green colors, for example, here, but also they're distributed. And there's like in the prefrontal cortex, as well as in the parietal cortex and temporal cortex, some representations of them. And again, bilaterally and across the 2 different experiments. Okay, so we can also study where visual and linguistic semantic information are represented in the brain. So we can move beyond only like language representation, linguistic representations, but also can look at whether when you watch a movie or when you just look at a scene, basically, the similar semantic representations, or like the brain is processing the semantic information similarly. Basically, because our naturalistic data sets are so rich, we can use the data that we collected also previously to do this. In this case, we used a data set that is collected by Shinji Nishimoto in 2011, and then also reused in another study by Alexander Hutz in 2012. And now what we do is basically we have subjects watch video clips. And while, so short video clips, these are silent movies. And each of the movie frame is then all the objects in the movie and the verbs and nouns are labeled separately. And you can see this in this manual labeled feature space. And then we are mapping this to the word embedding space that we are using in the story experiment to match it to the semantic features that we have basically from the story listening and reading experiment. And again, this is the same experiment that I mentioned in detail so far with the story listening from Hut et al. In 2016 and by myself in 2019. So when we look at this, we do the same pipeline. We use the model, the category, the new semantic features that are defining these movie frames, and create the voxelwise predictions for each voxel for this new semantic feature space in this data set. We can see that there is like this. Remember, this is the visual cortex here in the inner parts, the border to the anterior parts, the temporal lobe and parietal lobe. Here, this border is highly representing, highly predictive by the semantic model for the visual experiments. And here is again the experiment by when you listen to a story, and I am just flipping back and forth a little bit, that you can see there is a shift from the visual semantic representation towards more linguistic semantic representations towards the anterior regions. And a nice way to visualize this is these 2D maps that we use in these 2D color maps. Everything that is blue is well predicted by the semantic model for the language experiment, for the linguistic story experiment, and everything that is red is well predicted by the movie experiment. And everything that is more whitish is basically this border that I mentioned is predicted well by both of the models. And you can definitely see this clear border here, which was created by, which was studied very in detail by Sarah Popham in the lab, and that the movies are strongly represented mostly in the modality specific basically regions, whereas the stories are more represented, or the linguistic semantic representation is more represented in the high level anterior regions, such as temporal, parietal, and also prefrontal cortex. Okay, so in the last couple of minutes, I would like to mention some experiments that are trying to more bridge the gap that I would call between naturalistic and non-naturalistic experiments, right? We are doing these like very rich naturalistic cognitive experiments, but the majority of the field also is still, using a lot of like this classical fMRI condition-based, let's say, experiments. So we have several attempts that we try to bridge this gap between these two different experiments that I would like to highlight here. One of them is basically whether those contexts in language affects semantic representations as well. As I mentioned in the beginning, the classical fMRI experiments in language processing have been mainly on out of context word presentations to the subject, and then looking at the brain activity while while subjects are reading or listening to single isolated words. So we also wanted to study this and try to understand how this affects brain representations on semantic representations in particular. So we designed an experiment where we showed subjects, we took the same narratives that we had, and we just showed subjects words in isolated randomized orders. We also created two different similar conditions where we call them semantic blocks. The semantic blocks condition is basically words are again in isolated, shown in isolated out of context. However, we created or we grouped some words that are semantically related in small blocks. And then we have the sentences condition where we basically just randomize the order of the narrative stories. And again, we use the same pipeline, right? We record brain activity, we extract semantic features, we estimated some model weights, we use those model weights that we estimated to make model predictions. And when we have the predicted activity, we look how well we did on the recorded brain activity on the test data set. So when we do this, you have seen this earlier, basically. This is when you read the narrative story, words are temporally organized, there's a lot of context on natural language, and your brain is extremely interested, and we have this large pattern of bilateral temporal, parietal, and prefrontal cortex that is representing semantic information. However, when you start taking everything out of context and you show only isolated words to your subjects, and the brain gets very bored, and you can see that also when you ask your subject basically afterwards how well they did. It's extremely boring and difficult even to sometimes pay attention to these isolated words. So this shows how much it matters for the brain as well, to the brain, to see stimulus or words in this case in their natural context. We gain a little bit more information when we use words again in semantically grouped blocks. So those are basically, to give you an example, like apple, fruit, market, buy, table, knife, right? Things that are usually used in similar contexts, and you can see your brain is again more alert, you could say, and then you have more activity that you can predict for this type of condition. And if you have everything in sentences, which is used also a lot in the field, basically, the isolated sentences, we can see that those contextual information that is within a sentence already matters a lot more to the brain than single isolated words. And this is just the summary of it, single words, and then you are going counterclockwise in this case, no, clockwise actually, here to the narrative condition. And again, this was just to bridge a little bit this gap between how is narratives, listening to narratives relates to different conditions such as isolated single words. Another example to this is from Mark Lescroart, who is also in the audience today and going to lead part of the tutorial, is basically the work on intermediate visual representation in the brain. So Mark was interested in, or is interested in, whether scene selective regions are also representing information about 3D structure. And he designed basically a very nice experiment by creating computer-generated videos with, I think, over 800 scenes. And the beauty about this computer-generated videos in this case is basically that we know then the ground truth that we could use to extract different specific features, 2D features as well as 3D features from the movie clips. And using these feature spaces and then the Voxelwise modeling, he basically showed that scene selective regions such as here PPA, RSC, and OPA are representing the structural model, the 3D structural model better than, for example, 2D models. And he also showed this in a very variance partitioning analysis, that those models are actually explaining more variance than, for example, the 2D models. The last bit is a platform which is called boldpredictions.gallantlab.org. You can go to that link and see how we actually simulate classical fMRI experiments, currently only for the language experiments. However, the creator of this platform is Leila Vehbe, who is also extending this currently for the vision experiments, that is using basically our predictive models and our naturalistic experiments, the data that we have from, in this case, the story experiment, to make or like simulate whatever contrast you are entering into your into your experiment or into your question that you are asking in this platform. So just give you a little bit of an overview how this looks like. We have two different ways. We have the possibility where you can enter your own contrast, or we have some replications or simulated replications of published experiments. I think this is a video exactly. So you can enter a contrast if you want yourself. If you're interested in some new contrast that you would like to study, you can just go ahead and do it here, or you can just play around a little bit to go for the suggestions. And we have, like, for example, we can say sight words or smell words, sight words and sound words in this case. And then once you select that, you'll see that these are the words that are used usually in the literature to study this type of contrast studies, where you have different words. And then we are basically using, as I mentioned, the models and the data that we have in the lab from the story listening and reading experiment to make the predictions for these different contrasting conditions. Everything is done in PyCortex, so you can really use PyCortex to, all the features that PyCortex allows you, and, change the view of the subjects and analyze it, or like look into the data and visualize it for individual subjects, as well as actually for the MNI template. Again, here's just the view for that you can do with PyCortex very nicely with the flattens, etc. Okay, so the second part is, I'm not going to go into the detail this here, but we have the published experiments. We collect a lot of metadata for these experiments from the papers. We JSONize them and then include this into the analysis. So this is a growing part. But if you can see here, for example, just one example that I mentioned in the beginning with the concrete versus abstract words from the Binder study, this in the back, you can see the predictions, the simulated predictions that we create if we would use the concrete and abstract words that were used in Binder's study. You can see, and then the green hatches are those that are basically reported in the paper with the voxel locations and the MNI space. So this is the MNI template brain, and you can see there is like a matching with the green hatches is matching with some of the red portions, which is the concrete abstract concrete words representations simulated from the bold predictions platform. But we see also more activity. And some of them are not matching directly with the predictions or with the main data reported from the Binder study. And there is statistical analysis in the paper about this as well that is trying to figure out what, how reliable is basically the predictions and the matching conditions from the original studies. Okay, so I mentioned this in the beginning. Encoding models can be used to create accurate brain decoding. I think a lot of you in the audience know already this study by Shinji Nishimoto. He showed subjects these movie clips that I mentioned earlier in the visual semantics study to first create an encoding model based on the low-level visual information, and then use this encoding model with some natural movie priors in addition to that to reconstruct brain activity to see what movie clip the subject was basically seen. And you can see that your construction is obviously pretty remarkable. However, what I would like to mention is that this is many times also confused, even with the reviews that we get, that Voxelwise modeling is not decoding. It's much more than decoding. It can be used like to truly understand what information is represented in the brain, in particular voxels. And as an extension, once you have a good, accurate encoding model, you can create a decoding algorithm to reconstruct, for example, information directly from brain activity. And this review by Thomas Nazalaris is I think, greatly describing the subtleties. So I highly recommend everyone to read this. Okay, to recap, Voxelwise modeling, the steps that I mentioned in different studies, is a naturalistic framework for fMRI. We try to sample the real world as closely as possible by using the most rich naturalistic stimuli that we can use to answer a cognitive function, basically. And then we measure brain activity, and the high quality of the data is extremely important. Data is essential, basically. If our data is bad, our predictions, everything on the road will be bad, our models will be bad. So we spend a lot of time in piloting in one subject until we really get the best data quality, as well as with the same, what stimulus we will use. So this takes a lot of time usually in the lab. And it also involves, which probably in the tutorial will be mentioned as well, involves pulse sequence development in the lab by working closely with MR physicists. Then we extract features. These are basically the quantitative descriptions of the questions using those feature spaces that we either borrow from different fields, such as natural language processing that I mentioned with the word embeddings, or we design them ourselves. Can be manual, can be other computational models, such as the spatial temporal Gabor filters that Shinji developed. And this is basically the other part that takes a lot of our times in the data analysis procedure. Once we have the feature spaces at great quality data, we can start explaining the measured data using regularized regression. We treat this as a regression problem, as I mentioned, and then predict and generalize always to unseen visual scenes, or like language stimulus, and also individuals. And we always do this by splitting our data to different data sets for training, validation, and testing. And again, I can't mention it enough, everything is in individual subjects always. To conclude, Voxelwise modeling, using Voxelwise modeling, we can answer a lot of questions, several different questions using the same data set about not only where, but also what information is represented in the human brain. And this usually, this leads to actually robust results that can be used, that can be tested using different methods. For example, in the language domain, we can use different natural language processing algorithms that are, popping up currently every hour, basically, to study how well robust our models are in comparison to what we have, for example, published. Again, it's a generalizable, and we always reproduce our results for different individual subjects and also in different experiments. With that, I would like to thank all the Gallant Lab members, for the organizers here, and everyone else in the audience. Thank you for your attention, and I'm happy to answer questions.
I'll applaud on behalf of everybody. Thank you very much. This was a really fantastic talk. Thank you for sharing such an exciting method with us. I personally am really excited because now we actually get to learn how to do it too. So you don't get to just see what it can do and the power of this technique and all of the techniques related to it, but we can actually see how to do it, get our hands dirty. So to kick that off, we've had actually a little bit of a conversation among the panelists, which unfortunately the panelist chat is not visible to the rest of the audience, but we also have a Q&A, a submission from Afra Wolfschlager, who is asking a similar question. So why don't we go ahead and do that? And then we can also have a little bit of a panel discussion maybe among you and Jack and any of the folks from the tutorials who also want to get involved, and maybe we can have that lead then into the tutorial. So shall we do that? Shall we move on to kind of a Q&A here? Do you want to keep your slides up or do you want to pull them down? Yeah, so the first question, great. Let me switch this to gallery so everyone can be seen. Fantastic. So the first question is, thanks for the great talk. Sorry, I missed, how the feature regressors are created and how they account for bold response delay. Could you explain? And so Jack is answering in the Q&A, and so everybody should be able to see this answer. But we also had some similar conversation going between Jack and Gunnar, and so I will let you guys talk about what you were talking about in the chat. There was too much for me to follow.
Yeah, I think Jack already answered that in the discussion. Yeah, I didn't mention this, but this will be covered in detail in the tutorial part. We basically use an FIR filter on the stimulus features to account for that delay of the hemodynamic response delay before we fit the models.
Yeah, there's generally two questions that come up. The first is in this work. The first is, how do you come up with feature spaces? And then the second is, how do you actually do it? And the second part, how do you actually fit the models? Well, that's the whole point of the tutorial. And so we'll be covering that in great detail. The first question of how do you come up with features, that's more of an open-ended issue. I mean, how you come up with features has exactly the same problem as how you come up with hypotheses. It's kind of inevitably a combination of induction and deduction. Then when you want to come up with a feature space, usually the first thing you do is build it based on some theory or hypothesis you have about how the brain works. And in fact, you can think of a Voxelwise model as an instantiation of an explicit computational model of some hypothesis. And different feature spaces are essentially different hypotheses that all compete to explain the data that you have. So if you have a theory, then you can instantiate a feature space that reflects that theory. If you don't have a theory, then you're going to be doing something that's very, very inductive and probably more akin to something in machine learning, like you could fit an artificial neural network. The problem with doing that is that that may fit very well. You may be able to generate features that you can fit in Voxelwise modeling, but then you'll still have the interpretation problem. You won't really know why it fit, and you'll have to sort of do an in silico analysis to try to determine why the features that you didn't understand fit well. If you have an explicit model going into things, if you have a hypothesis, then the minute you fit them and get good predictions, you can immediately interpret what happened, and the process is a lot easier. And the last thing I want to mention on this is not an unusual problem. Every person who builds an experiment has to make the same kind of decisions that we make when we design a feature space. No matter what your theory is, if you're going to do an experiment to test that theory, you have to operationalize all of the variables in the theory into some parameters that you can manipulate in the experiment. And that process is essentially imposing a feature space on the world that you're going to use to sample the data in your experiment. So you're essentially selecting the subspace of the world that you're going to explore in the classical approach before you do the experiment and before you analyze the data. In this approach, you can use a much broader, more naturalistic stimulus and parameter set, and then impose structure on it after you do the experiment rather than before.
Can I ask, Jack, how you do that, in particular for the movie sequences that you showed, where it's not, in a way you have to infer, oh, now the participant is trying to make a decision in the driving, for example, whether you turn left or right, or is there a way to automate that so that you don't have to have too much sort of experimental controlled biases brought in?
I think if you're exploring, if you're trying to create feature spaces for very deeply buried latent variables, you're always going to have a problem. It doesn't matter whether it's driving, or imagine that Fatma's language experiment had actually involved a conversation where you have two speakers. So at a given point in time, the one person, even if they're not speaking, they could be listening to the other speaker, or they could be trying to formulate their response to that speaker, right? And those things are probably mixed up. So there are all these latent variables that are represented in the brain that don't have any direct behavioral correlate. And that's a tricky problem for everybody. And the classic experimental approach can't really do anything about that to address it. And this Voxelwise modeling approach can't do anything directly to address it either. But what you can do is create latent variable state space models that can be fit to the data. For example, you could use an MCMC model, or you could use some sort of game theory model. And then inside that framework, you can find the optimal parameters for that framework in your data set, and then predict and get an estimate of significance and accuracy. So I think that's actually, even though it seems like a problem like that would be impossible to study with Voxelwise modeling, it's actually easier to study with Voxelwise modeling because the computational framework you're going to need to express those latent variables and to model them is going to be built into the whole data analysis and modeling procedure. It's not going to be, you're not going to basically be trying to fit the square peg of the naturalistic task into the round hole of some really limited decision space in order to try to apply the classical approach. But I wish I had happier news for you. It's a good question and a hard problem.
Can I piggyback onto that answer with just one more sort of practical consideration? The other part of this was, how do we define these feature spaces? So they can be labels, they can be human annotations of whatever the stimulus is. And we're going to show you one example of that, which are WordNet labels for these movies. So what semantic things actually occurred, people in the Gallant lab actually went through second by second and hand labeled a bunch of WordNet categories in the frames of that movie. So that's not any kind of deep neural network. That's just a sort of intense manual labor kind of model. There are also possible pixel-based models to create, where you pass the pixels of the frames through a deep neural network or whatever your favorite algorithm is to derive the features that are there. And the third possibility is if you've got some sort of generative parameters of your stimulus, like for example, the blender stimuli or that driving experiment, it's possible to have either features that help to generate the stimulus or features that you can compute for that stimulus, like depth maps in the video game or in the blender studies, and then use those directly to model responses as well, which aren't exactly computed. They're just sort of part of how the stimulus was generated. So it's really, it's a creative problem. It is the creative problem in designing these things, is what should the hypothesis be? How should the ideas of a theory be instantiated through any of those methods that I just went through? And that's the art of any experimental design. And I do want to add one more thing, which is that even with naturalistic stimuli, you are sampling the world in a very specific way. You've chosen that this is the world, and the movies that we have perhaps are a better approximation of real human visual experience than, objects floating on gray backgrounds. But they have their limits, and any stimulus set should be assessed in terms of sort of in what dimensions does it contain variance. And what should those variances be to really reflect natural experiences? That's a really challenging problem, and one that my lab is currently working on, but that's a whole other story that I can talk about later if anyone's curious.
Great. Thanks for the really detailed answer. We have one other question before I think we move on to the tutorial, but this is a reminder also, please, everybody stick your questions in that Q&A box also throughout the tutorial if you have them. And so this second question is about talking about a little bit more about the ways you can relate the results across participants beyond the qualitative similarity.
Okay, so I can take that probably. So I briefly mentioned this in the talk. One way we can more quantify this is basically, as I mentioned, you can directly map the Voxelwise modeling, either the brain data onto the MNI space and do the analysis on the MNI space, or the results onto the MNI space, the template space. And then one simple, maybe I show that slide again actually, but if I have it open, let me open that again. What you can do once you do that is you can really count even, right, for each voxel, how many of the subjects were predicted significantly, right? And that is something that I showed. Sorry, I'm having a hard time with my mouse here. This one. So I glimpsed over a little bit on this just to show you that we can do this without explaining maybe more in detail. What is happening is basically for each voxel, I'm counting how many of the subjects are significantly predicting you know, the voxel activity for the semantic model, for example, right? And this is one way to quantify the prediction accuracies. If you're asking how you can quantify, for example, the colorful semantic principal component maps that I showed, these ones, you can really simply, and this is something that I did also, is basically you can, once you have your voxels in this principal component space, you can, for each principal component, correlate, right, between let's say listening and reading for each individual subject, and then look at how this performs across subjects as well, right? How the similarity is different across from the mean, for example. And this is another way to quantify these more, you know, interpretation related semantic maps. I hope that answers the question. If not, I can go into more detail.
Can I just add one thing that you briefly mentioned, Fatma? That
sure.
Basically there's nothing intrinsic in Voxelwise modeling that doesn't let you fit these models in a template. So for example, you could transform your data to a template surface, for example, if it's average, and run your models there. We generally don't do that because Voxelwise modeling is so sensitive that it allows us to get at finer distinctions in individual subjects' brain. And whenever you do, you know, an anatomical projection to a template, you lose all these high frequency information in the spatial patterns. So you can do that if you want, if your experiment is looking at that, but we generally prefer to fit the model first in individual subjects, and then only later project the prediction accuracies, as Fatma has shown, or by looking at group PCs to look at it across subjects.
Can I ask a follow-up question? So how do you interpret the not ideal cases where like only a subset of participants showed significance, for example, or when you just look at a single participant where, you know, the predictability is sort of like not like super high, let's say. And of course, like I understand that this is still a super useful tool for model comparison or hypothesis comparison and things like that. But I was wondering whether there was more that can be said, or more caution, or if there, because like, can you give me an intuition of how far I can go in the interpretation, or where I have to be cautious, and what to do with these cases where, you know, half the participants show significance, the other ones don't? What do I do with that? How do I interpret that?
Well, some part of that is actually individual variability, right? And we are interested in that, but it's a hard problem. And we are looking into that in different populations. I can just give you from my own experience. I'm currently doing a bilingualism experiment as well. And exactly those things are the interesting parts, right? What is the individual differences that some, let's say, bilingual person is experiencing when they read a story, for example, right? But it is, it's not an easy problem. So for now, we are usually, you know, we are preserving those individual variability in our maps. That's why we never really, you know, show necessarily as a first main results are template brain results, because that's something that says, okay, across these people, the pattern that holds. But we are always preserving the individual variability in the different maps. However, you know, again, like quantifying those are not the most simple, easy problem currently for any of the experiments that we have. What is the interesting part?
Yeah, I would add, Matteo's been working on this problem quite a lot lately, and I think he's going to have a paper soon on this issue. The variance due to individual differences is actually the majority of the variance. The variance due to the group model is a minority of the variance. So the individual difference is going to be very large. In our lab, we traditionally do small n studies because we are kind of doing more exploratory research, usually in areas people really haven't looked at before. I think, Gunnar, if you wanted to do the kind of if you want to look at the kind of issue you're talking about, which is what are these individual differences and how do I, how can I understand them? What you need is enough individuals that you can make groups of, you can make subgroups within the larger group. And that means you need a larger study, but it should be, I would argue it can't be, it shouldn't be a resting state study. It should be something that involves variables, probably complicated naturalistic tasks. And there are data sets like that coming out. Pierre Bellet's group is coming out with something like that. The folks at Neurospin have a big project Korean group is working on a big project. All of those are aimed at looking at individual differences in complicated naturalistic conditions. And those will be great platforms for applying Voxelwise model to individuals at a large enough scale that you can look at individual differences and actually find subgroups in order to help you be able to interpret those individual differences.
I can add to that. There's also sort of trivial individual differences, which are differences in signal quality. So the first thing that we're going to do in the tutorials is show you how to compute repeatability or a noise ceiling for every voxel. And so this is something that is part of most of our experiments. It's not always possible because it's not always possible to collect repeats of the stimulus that you're interested in. If you're specifically interested in, for example, the first exposure to a story and whatever your emotional reaction was to that, and if you think that that really might differ in subsequent presentations of the story, or if you have something like the driving experiment where it's, it's sort of governed by the way that the subject interacts with the video game, then you can't necessarily collect repeats. But to the extent that you can quantify to what degree every voxel is actually driven by the stimulus, and you can exclude voxels from your analysis that just aren't driven because of magnetic artifacts, because of physiology, because of whatever else. And that's a really crucial element of what we do, and it gives you a picture of the noise in your experiment at a voxel by voxel level, which I think for approaches that are purely multivariate, I think that's a little more problematic to know how noise might be affecting your stimulus or your conclusions in your results.
In fact, the most common MRI method used today is functional connectivity, and it has no way to separate signal from noise. So you just glue it all together and call it signal. It's that reduces sensitivity a lot, especially given the very, very large individual differences in signal to noise.
Well, on that note, maybe we should go on and actually see how some of this magic is done. There are no more questions in the Q&A for now, so we can go ahead and move on to the tutorials. But this is a reminder again that if you have questions throughout the tutorials and so on, please put them in that Q&A box. That helps separate them from the chat. And yeah, let's just go ahead and go on to actually get our hands dirty. And so now we'll move on to the tutorial section, which is led by Tom Dupre and Matteo Visconti. Is that right? And Mark Lescroart, you're gonna hang back for a little bit here, I think.
I'm here to answer questions.
Okay. All right, great. So yeah, please take it away.
All right, thank you. So thanks for staying here for the tutorial. The idea of this tutorial is to go a bit more in a concrete fashion after this very rich presentation and discussion. This is a notepad that the address is here at the top, or here at a smaller version. And I suggest all of you to go to this page. We can also maybe copy this in the chat. And in this page, you will find useful links that you can just use to go through the tutorials. And there will be also some supporting text and figures. But more importantly, there will be a question section here where you can actually ask your question live during the tutorial. And Matteo and Mark, Fatma will try to answer your question while I'm doing the presentation. And after a time, Matteo will continue the tutorials and I will answer your question as well. So please go on this page. So these tutorials will be organized toward this project, which is called Voxelwise Tutorials. There is a GitHub repo where everything is open source and you can find everything. But the tutorials will be formed by Python scripts or Jupyter notebooks. They're exact copies of each other, but they are not rendered. So if you want the rendered version, you can go to the website here. And the website is a good resource if you just want to scroll through the tutorials or give someone else that is not here today a quick overview of the tutorials. But during this presentation, I'm going to go through the notebooks live, and you can do the same by just clicking the Open in Collab here link. And this will open a Google Colab notebook where you have all the different notebooks we're going to go through concatenated. And for those that do not know about Colab, Colab is a Google service to run Jupyter notebooks in the cloud for free. And you can just do that if you have a Google account. If you don't, you can either download everything from the GitHub repo here, or just follow my presentation, or even like go to the website where everything is rendered and described. So this is the main project that we are going to use. A large part of it is also based on Himalaya. Himalaya is another project that is a Python package that implements some ridge regression like models. So this is the package we are going to use to actually feed the model. It's following a scikit-learn API. So if you're familiar with the scikit-learn API, it's really useful, but it's more specific to voxel-wise encoding models than scikit-learn in different ways I'm going to present. And one of them is in particular that it has a GPU backend. So a GPU will make your model fitting much easier and faster. And so the good thing is that with Google Colab, we can actually use a GPU, and it will make the tutorial much faster to go through. So this is the general overview of the tutorial. I don't know if we will have time to go through all of the 5 notebooks, but the first one, we will briefly explain the noise ceiling, the explainable variance that we just mentioned. In a second notebook, we will actually fit a model using some semantic labels that were manually annotated. And in a third notebook, we will go a bit more in details, talking about the hemodynamic response function and the delays that are used to fit the Voxelwise model. Then we will fit a second model with a different feature space. The first one was using semantic labels. Here it uses low-level visual information. And if we have time, we'll actually compare these two in a joint model with both feature spaces simultaneously, which is pretty exciting. So this is a very short overview. Each of the 5 blocks are kind of independent, and we can run them just independently, but we will just go through sequentially in this presentation. So without further ado, I think I will just start. And remember, if you have questions, to go to this section and actually write your question. So to do that, there is a link at the top right to edit. You can just click it, and you don't even have to log in to actually type your question. So here, I think it will log me because I am logged, but it will open this interface where you have on the right the notepad, and on the left a markdown version of this. So you can just click to go through where you are and type your question. And you see it appears live on the notepad on the right, and it's really straightforward to use. So please type your question. You can test that, and we will try to answer that live. So if you have not done it yet, you can click on Open in Colab here and go through this page. So on the left, you will find a table of content where you can find all the different sections. All the different notebooks are concatenated, but they are independent. And the first one is just a way to set up Colab very quickly, and it should be pretty straightforward to do. So the first thing we are going to do is to change the environment in Colab to use the GPU so that the model fitting will be much faster. So this can be done here by clicking runtime, change runtime type, hardware accelerator, and GPU, and save. And once you've done that, now every time we are going to ask Himalaya to feed a model, it will automatically use a GPU, and it will be much faster. And then you have to actually download the data and install the dependencies. So I'm not going to describe all of that, but basically it will download everything. So you just uncomment everything here, and you can run each cell. If you don't know about this interface, you can run the cell by clicking here or using Control and Enter, and it will just run this. So this is just a warning. You can just say run anyway, and it will do everything. So this will download the dataset, install all the dependencies, and some dependencies to actually use PyCortex during this presentation as well. So you can leave it here. It takes a few minutes, but if you happen to actually make your notebook crash, which can happen if you run out of memory or various reasons, it should not take as long because it will not re-download everything. So if it happens, you can always restart your notebook here by interrupting, restarting, and if you have to do a factory reset, then you will have to re-download everything again. So I'm going to continue while it's doing that in the background. I'm going to run that as well. It's just some configuration for PyCortex. And here you have a cell that just resets the variables. This is because all the different notebooks are independent, and because in Google Colab you can have some memory issues, it's easier just to reset all the variables between the notebooks. You don't necessarily need to do that if you want to go through by yourself, but if you have some memory issue that you can find here, the RAM is pretty low for now, but can increase, and you can just reset and start each notebook fresh. So we need to run that to plot the figures, and we can directly jump to the explainable variance. So as we briefly discussed after the talk, a key concept in Voxelwise modeling is to estimate if your model is poor because you have not found the good features, or if it's poor because the experiment is poor. Maybe there is too much noise, maybe the experiment is not repeating the same brain responses, and so there is no way to actually model anything. So computing the explainable variance is a way to quantify that. And the idea of this is to separate the signal, what we measure, into two conceptual components. The first one is the part that will be dependent on the stimulus. So if you repeat the same experiment, you will repeat the same signal. And the second part is actually not repeatable, and we call this noise. So to use that, we actually do the repetition. We have, in some cases, opportunity to repeat the exact same experiment. So watching the same movie, listening to the same story. And if you can repeat the same experiment, we can compute an average response here. So let's say we have n repetition. We can just average on each voxel the response. And what we hope is that the stimulus-dependent signal will be consistent over the different repetition, and that the average will keep it, whereas the noise part will be averaged out. And this is the assumption. So obviously, the more repetition, the better. And this is a way for each voxel to quantify what would be the stimulus-dependent signal. And now that we have a stimulus-dependent signal, we can compute what is the variance associated with this stimulus-dependent signal compared to the overall signal. So here's the formula. It's a bit complicated because it has some bias correction, but all you need to know is that it's computing the variance of this average compared to the variance of the signal in each voxel independently. So this is a pretty critical part of Voxelwise modeling. You cannot do that for every experiment, but if you can, it's really powerful. It's sometimes called explainable variance. Sometimes it's called signal power or noise ceiling, but it's all the same concept. And you can read more in these references that you can find here. So we are going to actually compute it. So you can run it here. So click on the hidden cell here. This is just to give the path to the data and to specify a subject. In the data we gave you, we have only one subject, but in the future we will release more subjects, and you can try with other subjects as well. So we are going to run this as well. And here we load the data. So the data here is a NumPy array. So just a block of data with 10 repetition of the same stimulus. On 84,000 voxels that are the cortical voxels that were extracted from the experiment, and with 270 time points, so number of time samples in this test set. So from that, we are just going to call this function that implements everything for you. And it will collapse this information into only one score per voxel, the 84,000 voxels. And it will be the amount of signal you can hope to predict with the encoding model in each voxel. So to give a better understanding, we can take the best voxel, the voxel with the highest explainable variance. And here we see the time courses of these voxels. And each blue line is a different repetition of the same experiment. And the orange is the average. And you can see that on this particular voxel with a 0.71 explainable variance, the signal is extremely reliable. And so we can say that this is really driven by the stimulus, because every time we repeat the same experiment with the same stimulus, we get the same answer, the same response from the voxel. So here we can really hope to predict a lot of information. On the contrary, we can find the lowest explainable variance voxel, and we can see that this is actually really noisy, and we cannot hope to find anything. So if the Voxelwise modeling does not predict well here, it is not surprising. We cannot even hope to predict anything, because we will have the same features for each repetition, but the response will be different every time. So this is a voxel that we can immediately discard. Here, do not pay too much attention on the negative weights. It's due to the bias correction, so that if you have noise, it's centered around 0. But essentially, it's a low value. So to give actually a distribution over all the different voxels, we can plot here a histogram. Note that every time you have a cell, you can run it by yourself, and you can actually modify it. Here, for example, I start at 0, but I could have started to show negative values as well. So I can just run that again, and it will show up. So feel free to explore by yourself. This is the goal of this tutorial. And here you have a histogram of explainable variance, where you have in log scale, here's the number of voxels, and you see that a lot of voxels are actually centered on 0 explainable variance. So these are voxels we can ignore for the rest. But a large part of voxels are also having a pretty large explainable variance, up to 0.071, as we said. So from this result, we can say that our experiment is not completely bad, and we can hope to predict something. So obviously, this doesn't tell the full story. We want to show we want to know where these voxels are on the brain. So this is the next step. And the usual tool we use for that is called PyCortex. It is extremely useful to show either flat maps or to navigate on 3D viewers. We're going to open that next. But for some reason, like for some privacy reasons, we do not share all the anatomical scans that are necessary to open PyCortex. So instead, what we have is a mapping that goes directly from the voxel we gave you to the flat map. So you can just run this, and it will show directly the flat map. So this flat map is specific to the subject. We have all the ROIs labeled, and what we see is that the large explainable variants that are here in the purple, orange, and yellow are located in the visual cortex, early visual cortex, and more like semantic visual cortex, like ABA, FFA, the face area, the body part area, etc. And what we have from this is that this is what we were expecting, which is the visual, like the movie experiment we used, is triggering some pretty consistent response in the visual cortex. So this is specific to the subject. What we can do instead to use actually PyCortex and have a 3D viewers is to map this subject to fSaverage. fSaverage is a template space where we can map different subjects and they go into the same space. So we can just run these, and here it will download the subject. But because there is some issue with Colab, we need to reinitialize the file store. So don't focus too much on the details here. But this is the important part. It's downloading the surface. And now we can actually translate the organization of all the voxel into fSaverage space, like project with this projection. And now we have, instead of 84,000 voxels, we have 300,000 vertices. So each point on the fSaverage mesh. And we can use that with PyCortex to create an object, a vertex object that we will use for visualization. So I'm going a bit fast here because we will not use so much PyCortex here, but I really want to highlight that this is an extremely powerful tool. So this is actually a trick to make it work with the Colab. Usually when you want to open the PyCortex 3D viewers, you have a PyCortex installed locally on your computer, and it will use a local like server to actually show the brain viewer. But because we are running everything in the cloud, we need this small trick. It will give you an address where you will be able to connect to see the brain viewer. And now we can actually run the brain viewer. So you can change that to true. And it will generate a brain viewer that you can click here. So usually it gives you a link, a local link that you can use to open the brain viewer. But what you can do instead here is use this link because we have this issue with the running everything in the cloud. So I think I sometimes have an issue with this particular brain viewer, but I can actually show it on Chrome instead. So if you have some issues, you might want to try a different, oops, it is not working. So this is a problem with live demo. All right, so I'm sorry about that. I can try maybe here. Okay, so there is an issue with that. Maybe it's using the same ngrok link for, I don't know exactly what's going on here. But basically this would was supposed to show a brain viewer that you can also find. You can find some demo online on the PyCortex. The idea of the brain viewer is that it's a 3D viewer that you can navigate and explore the brain in 3D. So I'm sorry.
Sorry to interrupt you. I'm following along. Would you like me to just show it? Because it did work on my machine.
Oh yeah, please do.
Okay, just so that everyone can kind of see what this
that's awesome.
What this looks like.
All right.
So it looks like this. It's very pretty.
So you can turn around and see everything. What you can do as well is press H, which will give you a number of key to press to change this view. So what you can do, for example, is press F to flatten the brain, and this is the usual view that we have in the presentation, because it is a useful way to show the entire brain at the same time. But if you want to go back to the 3D view, you can just press R to reset the view, and it will recreate the brain if you are more familiar with this view. So there is also a lot of controls if you open the console on the right. On the top right, you have, for example, if you click on surface, you can have a lot of controls to change this view. I'm not going to go too much here in detail, but I highly recommend this tool to explore the different maps you have created from the brain. So I'm going to go back to the tutorial now. And if it doesn't work for you, it's not a problem, because we will not use this PyCortex too much. We will directly use the mappers that, so if you have these already working, we will use this kind of plot every time. So don't worry too much.
May I interrupt with one more thing here, Tom?
Of course.
There's a chance that you also might not have been able to download the data. We have two popularity problems. One is this tunnel for PyCortex, and the other is access to the data, all of which is being done through Google Drive right now. And we seem to have hit a maximum for a number of requests for both of those. So it may just mean if you want to run this, you may need to run it again tomorrow.
Oh yeah, okay. I'm sorry about that. We did not anticipate this problem. If you want to also look at the different figures that are generated, because maybe I'm going too fast on some of them, you can go to the website. So here you have the website link where you can find the tutorials. So if I go to compute the explainable variance, for example, you will have the same notebook with everything rendered already. And you can, obviously you cannot play around with this by yourself, but you can still look at the figures and look at the different description of these figures as well. And here is, for example, the kind of plot you get for the FS average. Actually, I'm going to run it myself here. And this is just to show the FS average surface. So don't worry about this. And this is a different flat map compared to the previous one. which was specific to one subject. But it's essentially showing the same information, that the explainable variance is in the early visual cortex. So yeah, so this is the end of the first notebook about plotting the explainable variance. This is a key concept because now we know where we can hope to predict something, where if we do not predict something, is it due to the modeling part being not as good, or is it due to the experiment being not as good? Here we have already disentangled a bit of that. If you want to read more, you can find these references about it. And if you have some issues with running the notebook, you can just rerun this part and jump directly to the second notebook, skip everything, because all the different notebooks are independent. So just to ensure that we can actually reset everything and start the second notebook. So in the second notebook, we are going to actually fit the ridge model. So ridge is a linear regression method that regularize the linear regression to be more stable for various reasons. It's much more powerful than just linear regression, in the sense that you can, for example, fit a lot of different features and not have a problem of collinearity that you have in linear regression. So here, the WordNet features that we are going to use are the one that we already described earlier. So if we take a look at this figure, we have the movie and a set of labels that were manually labeled on each second of the movie. We actually labeled if there is a woman, a man, a building. Maybe there's no dog or no car, so we can put a 0 on these two categories. But we did that for a lot of categories. Actually, we did that for 1,700 categories. And based on that, we are going to feed a model that goes from these labels to the brain activity. So this involves a number of steps, but hopefully this tutorial will show you that you can do that by yourself and also try on your own data someday. So we're going to load the data again. Here we have two sets of file, the train set and the test set. So this is, I think, extremely important, and I will emphasize a lot on that by going back to this figure. Here we have two different, for example, feature spaces that we want to try. But if the more complex the feature space, the more you can explain something on the training set, and it will maybe not generalize to a new data set. So to ensure that what we estimate is the prediction, the correct prediction accuracy, and not the overfitted prediction accuracy, what we do is we train the linear regression on the test set. So from X, the input, here the labels, to Y, the brain activity, we estimate a set of weights B here that will be used in the linear regression. And then we test it on a different set. This is critical. We use the features and the estimated weights to predict some brain activity. And because we have recorded brain activity on the test set, we can compare with the ground truth activity and get an evaluation of the prediction accuracy. Here, we'll use R². We can use other prediction accuracy metric as well. So if we go back to this, we have two different sets of data. So Y train the responses on the train set, and Y test on the test set. And remember that we have here 10 repetition of the same. So we are going to average them to get the stimulus-dependent signal. And we are going to fix any potential issue with northern embers that might arise. And here we are going to actually load the features. So we are not going to go through the process of creating these features. This is actually complicated and time consuming. We just give you the features for the train set and the test set. And in another feature spaces that we are going to use in another notebook, we use motion energy, and there is actually a notebook that you can find on the website to actually create this feature space. But because it's quite long, I'm going to just give you here the pre-processed features. So we see the same number of time samples on the train set, on the test set, and here 1705 different features that we're going to use to predict the brain activity. So before we actually write the model, we need something else, which is the cross-validation scheme. So because we use so many features, we need a regularized regression that will actually improve the prediction quite a lot. But this regularized regression called RIDGE use a hyperparameter, and this hyperparameter needs to be set. And if we estimate this, the way to actually estimate it is to use cross-validation. So we will use the same idea here that where we splitted the data between train and test. We will do the same, but within the train set, we will split the data between a train and a validation set within this set, and we will use that to fit a ridge regression, test it on the validation set, and compare the different prediction accuracy and for a number of hyperparameter, and then select the hyperparameter that is the best. One thing that we have to do, though, is that because we are splitting this train set into a train and validation, we have to care about the way to split it. Because if we consider the different time samples in the brain responses, because they are correlated, we will artificially inflate the generalization, because we are going to train on one time sample and validate on the next one, and it will work too well, actually, because they are very similar. So a way to avoid that is to generate a cross-validation scheme that separate, that do not put one time sample in the train set and one time sample in the validation set. So what we are going to do is to leave one run out. So we are going to train on all the runs but one, and validate on the last one, and do that for several several selection of the out run. So here we just load the run length in the training set, so that we can generate this cross-validation split. So here, to give you an idea of what this function is, we can actually show what it returns. It's just returning a set of indices to be used in the training set and in the validation set. So now we need to define the model pipeline. And because we use Himalaya and that it follows a scikit-learn API, we can use a scikit-learn pipeline. So we're going to see that in a second, but basically we just need to define a number of steps that will be used in the pipeline. And these steps, the first step that we are going to use is a normalization step. So this is here called standard scalar in scikit-learn. And this is a way to remove the mean and potentially divide by the standard deviation of each feature. This is important because we are not going to predict a mean value of each voxel, because they are not informative in fMRI recordings because actually there is a detrending in the preprocessing of fMRI of ball responses. So the mean doesn't have any information in it, and we remove it. But because of that, we need also to remove the mean of the features. So this is what we do here. And you can see that we have with mean equal true, which means that we are going to normalize by the mean, but we are not going to normalize by the standard deviation. This is it makes look like a detail here, but the WordNet features are one hot encoding, meaning it's at each time point we have either 0 or 1. And if we normalize by the standard deviation, we will scale disproportionately the features that are very infrequent, which have very few ones, and most values are zeros. And here you might think, okay, I trust you on this one, but how can I know that it's a fair decision? And one good thing with the encoding model framework is that every of these modeling decision, you can actually test them in the sense that I could rerun the same analysis I'm going to run with STD equal true and compare the two different decisions with or without the normalization, and take the one that leads to the highest prediction accuracy. And this is a general idea of the Voxelwise encoding model that legitimize every kind of decision you have to make when you're doing this modeling part. So here I'm going to use false, but you can try by yourself and see that it will lead to poor prediction accuracy. Another step we're going to use in the pipeline is a delayer. And I'm not going to go too much in detail because Matteo will describe that a bit further in the next notebook. But the idea is that the brain response that we measure, the bold response, is delayed with respect to the stimulus. So if you if you see a car, the brain response for the car will happen actually a few seconds later. And because of that, we have to introduce a delay between the features and the brain responses. And we actually introduce multiple delays to improve the strength of the prediction model. Here again, you can trust me that 1, 2, 3, 4, meaning that we copy each feature and delay with 1 time sample, 2 time sample, and 3 time sample, etc. These values are good in this particular example, but you should not trust me on this one, and you should maybe test different numbers here, maybe going only to 3, or maybe actually going to negative delays. You can test that and keep the decision that led to the highest prediction accuracy. So I'm going to use that here. And finally, we're going to use a ridge model. And here you can, there's a bit of discussion why we use Himalaya instead of scikit-learn, but Himalaya will be much faster and give you better handling of the voxel-wise part of the modeling. So doing everything on each voxel independently. And another thing that, so here we are going to not use ridge, but kernel ridge CV. So CV means the cross-validation is done internally, and it will just compute the hyperparameter automatically, given the split we have defined earlier. And the kernel part is just a linear algebra trick to make it faster. You should not care too much about that because it's just a computational trick. It will give the same answers because we use a linear kernel, so it will just be a linear regression as well, but it will be faster. So last but not least trick to make it fast, we are going to set Himalaya to use a GPU. So Himalaya is a can use different computational backend when fitting the model. And the way to set them is just to use set backend. And the different backends that exist are NumPy, which is the default one, which will do everything in CPU. But you can also use PyTorch or QPy to do the computation on GPU. So depending on what you have installed, one version should be easier to use, but essentially in the back, the result will be exactly the same. So if at this point, maybe you have an error that says that GPU are not available, this is maybe because you have not used runtime, change runtime type, and GPU. Unfortunately, if you need to do it right now, you will need to rerun the different steps before because it resets the notebook. But if you have done that, the GPU setting, you will have this answer saying that it has loaded the GPU correctly. So we're going to use single precision numbers to make the most of GPU, because this is how GPU are fast, is by using, they are optimized for single precision floats. And we're going to define the grid of alpha parameter. Alpha is the hyperparameter in the ridge regression that needs to be set over the cross-validation. So we're going to use that. And finally define the fitting object, so the model. So we have the different alpha. Here we give the cross-validation split we defined earlier, and some hyperparameters that are necessary to compute everything with a limited memory in GPU. So this was a lot of maybe to introduce before actually fitting the model. But to summarize, we can organize everything into a pipeline, a scikit-learn pipeline that we just give the different steps that are compatible with scikit-learn. And here we have an overview of the model we are going to use. So first, the standard scaler to remove the mean of the features for each feature, the delayer that will copy and delay the different features, and the kernel ridge CV, which will estimate the ridge model, doing a cross-validation, and estimating the best hyperparameter over it, and then actually feeding the linear regression to make the prediction. So if you are not familiar with scikit-learn, maybe it's a bit fast here. I suggest you to go to the scikit-learn website to find some tutorials if you maybe some time to get you familiar with the concept. But here I will assume that you know a bit of this API. Just to make it very fast, scikit-learn is a very standard API for fitting models, and the concept of fitting and predicting has really were generalized to a lot of packages in the machine learning community. So this is the standard way to think about models. And to show you how simple it is to use scikit-learn, once you have a model, either one model here or a pipeline that is essentially just a longer model, you can just run fit, and it will fit everything. So here under the hood, it will go through the pipeline for the features, standard, normalize them, delay them, and then use also the responses inside the kernel ridge CV to make the prediction. So here I want to insist that we use only the train set, so that every parameters that are optimized here and learned on this set are actually learned only on part of the data that we will not use for the prediction accuracy estimation, which will be on a different set of the test set. So I might be repeating myself a lot here, but I think this is a very critical point and one of the main strengths of the Voxelwise modeling framework. So this usually takes about a minute, and once it will be done, This is a minute only because it's GPU. If you use a CPU, it will take much more, maybe half an hour or something. And then what we will use is pipeline.score to do the prediction on the test set and compare with the recorded brain responses. So it will give a different score per voxel, which is also a small difference compared to scikit-learn, which usually return only one average value. Here you see 84,000 different scores. And because it returns something, a PyTorch object, because it was fed in on GPU, we need to move it back to CPU with this command. And now we have the prediction accuracy. So let's take a look at how it looks like on the flat map. So here again, we use a mapper to plot the result, but usually we would use a PyCortex, which has many more features to visualize this result. So here is the result, and what can we say about it? We can see that it predicts in some of the areas where we had explainable variance, but not all, especially not in the early visual cortex. But it makes sense because the features we used were not low-level visual features, but more like semantic high-level concepts about what the subject was viewing. And so it makes sense that we predict in areas that correspond to semantic visual concepts. So this is working pretty well. This actually has been published in 2012 by Alexander Huth and colleagues. And you can read more about this. There is a link, I think, in the tutorials or in the notepad. So this is already a success, but what we want now is to better understand these regions and not necessarily say we can predict them, but we want to interpret them. And because now we have a computational model, we can take a look at how the computational model is predicting these regions to interpret what's going on here. So to do that, what we are going to do is not look at one voxel in particular. You could just take one voxel and look at the weights of the linear regression and see which feature are actually used to predict this particular voxel. But to do that a bit more systematically on all the different voxels and on all the different features, what we will use is principal component analysis. So, oh, sorry, I'm going to skip that because that's in the interest of time. But let's visualize a regression coefficient. So this unfortunately takes a bit of time as well, maybe 1 minute. The idea is that because we have this linear trick to fit kernel ridge instead of ridge, we have to compute the linear regression based on the kernel linear regression. You can ignore that. It's just a linear algebra trick. But essentially, at the end, what we get is an array with all the features which have been delayed multiple times, like 4 times in this case, per voxel. So this is pretty big. We are going to not consider all the voxels here because a lot of them are just noise, as we saw in the explainable variance. We are going to just take a look at the ones that have a score that is, let's say, above 0.05. Here it's arbitrary, but it corresponds more or less to voxels that have a significant score. Actually, the significant score is lower than that, but we keep only the good voxels here. And here we have still a number of delays times number of features array. So we are going to split the different delays and features to average the different delayed features. Just to simplify here the interpretation, Matteo will show that you can actually take a look at the delays part a bit further. So now we have the selection of voxels, the 1700 features, and the 4 delays, but we average the 4 delays to get only 1700 features. And we can use that in a PCA. So PCA is a principal component analysis, is a way to reduce this very high dimensional matrix to something that preserves most of the information, but in a very much lower number of dimensions. Here we selected 4 because we wanted to reproduce the original paper that looks at 4 components. And here we can compute the explained variance of the PCA. So what it means here is that the first component of this decomposition accounts for 20% of the variance of all the different weights. So what it means is that with only one dimension, we already capture 20% of the different values that arise in the linear regression coefficients that we want to explore. So it means that taking a PC, took at the first PC, we are going to already have a pretty good big picture of what's happening. So there's some tricks we use here because during the labeling part, the labeler probably labeled something like wolf at some point, but did not necessarily labeled canine, carnivore, mammal, vertebrate, etc. And so because the labels we used are part of a hierarchy of labels, we can automatically assign the different labels to their different steps in the hierarchy to be able to better understand this principal component. So this can be done directly by this correct coefficient function. And after that, we just normalize it. And we can plot this first component. So what we are going to take a look at is the nodes. Here are the labels that were used, and the different connection between nodes are the semantic connection in terms of hierarchy. So for example, drag is a special instance of a more general move label. So this is one of the aspects of this WordNet hierarchy of labels, is this possibility to show this hierarchy. But here, each node is sized by the weights on the principal component, and the color is positive or negative. So red is positive, blue is negative. And what we can see is that already groups of similar semantic concepts are grouped together in the same component. And here we have the first component correspond to the concept of moving, of person, against some event of like movement, and in the opposite direction you have more like artifacts, objects, etc. And so this first component highlight a main axis of change in this set of linear regression weights. Now we can also project this principal component onto the brain to see where the red part is projected and where the blue part is projected, and see the corresponding brain regions. So I'm not going to describe all, basically what's happening here, but I'm just projecting on all the different voxels, what we have done only on a small selection. And what we get is this. And here we can see that the red part is located, for example, in MT, where we have more like, we know that it's a region that has been linked to movement. And we see that the red part is actually corresponding to also movement. And so it makes sense to have this link between the the hierarchy of labels and the brain projections. So I'm not going to comment too much on that. It's slightly different from what you get in the paper, the original paper. And the reason is that the pipeline is slightly different, especially here I'm analyzing only one subject compared to the original paper that analyzes the PCA over multiple subjects. But essentially, the approach is similar, of like projecting to PCA space and looking at the first component. And we can actually also look at more components. So we are going to take a look at the next 3 components and color this, because we are looking at 3 at the same time. We color them with RGB, red, green, blue. And here we have some representation of this first 3 components. And again, we can plot that on the flat map to get a sense of where the yellow or the pink is projected. And we get this kind of map. Again, it's different from what you get in the paper. I think I would trust more the one that is on the paper, because it has more subjects and it's more described that I'm going to do here. But essentially, the approach is the same. And what we can do from here is compare the different regions that are now much more detailed compared to this projection, to understand what they're extracting from the very high dimensional space. So here are the references. This is the paper that we are reproducing here. And I would like to now give the space to Matteo to go through the next notebooks. We are maybe doing a small break. If you have some questions, maybe Matteo can answer them live as well. And then we are going to go to the next notebook.
Sure. Thanks, Tom. Yeah, if you guys have any questions, feel free to write in the HackMD or in the chat here. I'm going to start sharing my screen in the meanwhile. Hope you guys can all see my screen.
Yep, looks great.
All right. So I'm just going to go ahead because we are short on time. And in the interest of time, I'm actually going to change gears. And Tom has already shown you how to basically fit a Voxelwise encoding model using Himalaya. And I must say, as a user, it's incredible how easy it is. We're super lucky that Tom is a scikit-learn developer, and he basically wrote this whole package to fit Voxelwise encoding models. And he has a paper coming up where, what he described as a linear algebra trick is actually something that allows us to fit very complicated models in 4 hours versus days on the GPU. And that's really remarkable, and definitely has made my life much easier. So what I'm going to do now, instead of running each cell, is just going to be to describe the key concepts that we're showing you in these tutorials. And I think this is going to be useful because you can run the tutorials on your own, or later when you watch the recording, you can match basically what I'm saying with what's in the tutorial. As Tom said, now in this third tutorial called Plot Hemodynamic Response, we're actually going to address the question that was raised earlier in the Q&A. That is, how does Voxelwise modeling address the delay in the bold response? And if you've ever run fMRI experiments, and if you've looked at fMRI data, you know that fMRI is not really measuring neural responses. We would really love to say that fMRI measures neural responses, but the reality is that the dynamics that link neural processes to bold signal are very complicated, and they're called in general neurovascular coupling. And they're not directly. We can't really say that both signal is neural activity. And one way that this reflects in the data that we acquire is that the onset of our activity in the both signal is delayed with respect to the onset of the actual stimulus. You know that if you have analyzed the fMRI data using classical GLM approaches, we often use a so-called canonical HRF, or hemodynamic response function, that is in general convolved to our design matrix to account for this delay. In general, this HRF is fixed. It was estimated, if I recall correctly, from early visual areas, and we kind of assume that that function is the same across the whole brain. Unfortunately, that is not the case, because the HRF changes depending on vascularity, maybe even depending on age, and so on, and other factors that we probably don't know. And so in Voxelwise modeling, we prefer to have a more data-driven approach, which is under this umbrella term called finite impulse response models. And practically, I'm just going to show you the practice of like what this means, is that we have a feature, we have our feature spaces, we just delay our features in time, and then we stack them. So we create this much larger matrix, and we fit our model with that feature space, which is what Tom had shown you earlier. Here in this tutorial, we're just going to go one step further and analyze or look at the effect of these delays. We're going to see that if we fit a model without delays, they actually perform much, much worse than a model with delays. And we're also going to be looking at exactly what the weights represent when we look at the different weights, at the different delays. And we'll see how, by using this trick or this theory, Voxelwise modeling can actually recover the shape of our HRF function, our HRF response in different brain areas. So again, here I'll go very quickly. We load the data as before. We mean the test data to get only the average across repetitions. And then we load again our feature space. In this case, we're still fitting our model on Wernet features. Very importantly, we define the cross-validation scheme as Tom mentioned earlier. We define the model. This is exactly the same as you've seen before, and we get to our model, to our pipeline, and we fit the model. Now, this is our model with delays. Before comparing it to a model without delays, I want to take a little bit of time to understand exactly what these delays are. And in general, the best way to understand delays or to understand anything that we do in fMRI or in neuroscience is to simulate it. So here, I'm just simulating very simply 100 seconds of bold activity, which is actually just random data. Here I'm saying I want 50 TRs with a repetition time or sampling frequency of 2 seconds. And I'm just creating activity of one single voxel called Y. And I'm creating a very simple feature X that has some activity that I decided, in this case 0.5, between 30 and 40 seconds. I say here 30 and 40 seconds because our TR is 2 seconds. And so the sample at 10:15 corresponds to 30 seconds. I add a little bit of noise just because it's good to add some noise in your simulations. And then we create this delayer object that we've been using before in our pipeline. But now we're just going to create it so that we can visualize exactly what it's doing. We instantiate it and then we fit transform it on our feature X. Now, if you've never seen scikit-learn transformers or APIs, this is very common. They usually have, they usually have this, if they're transformers, they have like a fit and a transform method, or some of them, they have the fit transform method that does both at the same time. What I want to highlight here is that I'm just creating one extra dimension. This is a NumPy shortcut, or if if you will, because most of these methods want a 2D array as an input. So here, my simulated feature is just one single vector. And so I'm making it a 2D array. Now with this very simple simulation, we can just visualize what we have simulated. Now I'm just going to show these two lines here. This red line is my simulated bold response. Again, it doesn't have any meaning. The shape is not meaningful, it's just random, but it's just to give you an intuitive sense of the entities that we are comparing. And here I have in black the feature, my simulated features. And you can see I have simulated this ramping, this peak here that goes from 20 seconds to 30, from 30 seconds to 40 seconds. Now this feature here, or this trace here is not delayed. So it's indicated as x of t minus 0 because it doesn't have any delay. What delaying the feature means under the FIR method is basically pushing this trace forward in time by how many samples we ask it to do. So for example, here at x of t minus 2, because we have a 2-second TR, this corresponds to our feature being delayed by 1 sample. And et cetera, et cetera. Here we did it up to 4 samples. So we're basically shifting in time our forward, our feature, so that when we're fitting a model, imagine that we are fitting our linear model here at time 40, we're not only using whatever feature we had at time 0, we're also using the feature or the value of the feature at time t minus 2, t minus 4, t minus 6, t minus 8. And this is basically how FIR or FIR methods work, and how Voxelwise modeling can account for the delay in the hemodynamic response. And at the end of the tutorial, we'll see that actually by doing this, we can actually recover the actual function or the actual shape of the HRF. Okay, now we can then compare our previously fitted model that was fitted with delays to a model without delays. And here, again, we define our pipeline, our model as a scikit-learn pipeline. But differently from the other model, we do not add a delayer. So you see that here the delayer is missing. We fit our pipeline as before, we compute the scores, and then now we're just comparing the scores that we got, the prediction accuracies that we got on the test set, with delays or without delays. And it's useful to visualize this on a 2D histogram, like in this case, where on the x-axis, we're looking at the model without delays, and the y-axis at the model with delays. And as you can see, the model with delays is actually strongly outperforming the model without delays. And this is because, again, this model with delays is able to better capture the shape of the signal that we're trying to model and the dynamics of the signal. Now, it's useful to basically visualize what these weights are to understand even understand even better what exactly these FIR methods do. And so what we're doing here in this cell is fitting a model with many more delays. As you can see here, we're using actually delays from 0 to 6. So these are samples. This means that we're using information in the features that goes from 0, so exactly at the onset of whatever was occurring in the visual stimulus, to 12 seconds before. And we're going to fit this model only on 10 voxels, just because we are here, we're just wanting to show a concept. I want to add that one could have the desire to basically say, well, if Voxelwise modeling can recover how many delays I want, then I can just add so many delays, and then it will even capture very strange shapes or HRFs. Okay, I run everything, so that's all right. So I was saying there could be this idea of using as many delays as we want. However, that's not advised because, again, we're basically adding more parameters to our model. And whenever we add more parameters, we risk the risk is overfitting. So again, this is, as Tom said, one of the benefits of Voxelwise modeling is that you can test your pipeline steps in a principled way, but you always want to be careful and avoid overfitting. And one way that Voxelwise modeling avoid overfittings is first by fitting the models in the training set, validating the models within the training set, and then computing generalization performance also in a different set, in a held out set, in the test set. However, when we are developing pipelines or analysis, what we tend to do is to split our subjects themselves into different data sets. If you will, one data set is the exploratory data set, where we try different analyses to see which work best. And then we fix this pipeline. We don't modify it, and we applied it as is on a different set of subjects, so that we basically have replications of our results at the individual level on the subjects, as Fatma and Jack have shown, but also in independent subjects. So here we fitted our model with more delays, and this code is basically then plotting for one feature. It's plotting basically the weights associated with one feature at separate delays for 10 different voxels. On the x-axis, you have the value associated with one of these delays. Again, we have from 0 to 6, corresponding to 0 seconds when the feature was present in the stimulus, to 6, that corresponds to 12 seconds before of whatever feature was present. And on the y-axis, we have the ridge coefficients, or what the model, or the fitted models. Sorry, the fitted model weights. And as you can see, the positive or negative is informative in a sense that it means that when the feature is present, the model predicts that brain activity will increase if the weight is positive, will decrease if the weight is negative, compared to a baseline. However, what you can notice here is that the weights correspond to this delay that we know exists in the pulse signal. So for example, for some voxels, the peak is at 2, which corresponds to 4 seconds. But interestingly, and this is one of the features of using the FIR method, is that you see that some other voxels, like this teal or light blue voxel, has a peak at 3 or 6 seconds. So with this flexibility, we can basically try to recover whatever shape the HRF has independently in different brain areas, and just let the data tell us what the best and the optimal way to fit this model is. So I hope that was not too fast, but I know that we have a recording and we have 20 minutes left. So I'm gonna continue with this pace. If there are any questions, also Fatma, please interrupt me if there's anything that should be covered. The next tutorial, the next notebook is actually very exciting because we're getting to another feature of Voxelwise modeling, which is model comparison. In the previous tutorials, in the previous models, we basically used only one feature space, which was WARNet, which for one we can call it a semantic feature space that describes the semantic content of the stimuli. Here we're using a different feature space called motion energy that was originally described in 2 papers by Nishimoto et al. In 2011. One was based, when it was developed on animal neurophysiology, and the other, and then the same model was then applied to human fMRI data. And this model is basically quantifying with banks of spatiotemporal Gabor filters. It's quantifying the amount of motion or motion energy at different scales in your visual system, in the visual percept, in this case, in the movies. And what these motion energy features do, they're really, really good at capturing low-level image information that varies over time and over space. So what we're going to be doing here in this tutorial, we're going to be comparing the prediction accuracy of this type of model, the motion energy feature, that if you want is a low-level visual feature space, to a higher level semantic feature space like warning. So again, here we're setting up our data directory, loading the data. Again, everything is the same as you've seen before. The only difference here is that if you want to load the motion energy features, you just need to specify that you want the motion energy features and not the WordNet. Tom mentioned it briefly, but if after the tutorial you go on the website or on the GitHub repository, there is a tutorial where we actually compute these features. They take a long time. They take about 1 to 2 hours for this particular stimulus. And they are, I just want to mention that they are computed using this other package that the Gallant lab developed called PyMoten, or Python Motion Energy, that basically allows you to estimate these filters, or to estimate these features based on any visual stimulus that you have. So when you're running Voxelwise encoding and you have a visual stimulus, this is a very quick way to have a very good low-level visual feature space that you can use to compare whatever other feature space you're interested in. Again, we're defining our consolidation scheme. We define the model. Again, this is exactly as before. The only difference is that we're fitting our model on a different feature space. In this case, X-train is not the WordNet feature, but it's the motion energy feature. And you can see that also here. I think I have it. Yeah, you can see that the number of features for this feature space is much larger. So it's almost, let's say it's 6,500 features versus 1,700 that were the features of the semantic feature space or the WordNet feature space. Now, when we look at the prediction accuracy on the test set, and if you compare it to the WordNet feature space, you can see that this model is predicting much, much better in early visual areas as compared to the WordNet feature space, as expected, because this is a very good model of visual processing. But it's also predicting in some, let's say, visual semantic areas, like, for example, the fusiform face area, the extra-striate body area, etc., etc. Now we can actually compare it directly by here we're refitting the WordNet model. Again, the code is the same. The only difference is that now we're loading the WordNet features. And here we can see the same results that Tom showed before, where basically this semantic feature space is predicting more anteriorly than V1, V2, V3, the canonical early visual areas. When we have this type of models, or this type of model comparison, it's often very useful to look at 2D histograms and 2D flat maps. 2D histograms are, we've seen a bunch of them already before. And here we're plotting on the x-axis the prediction accuracy on the semantic WordNet model, and on the y-axis the prediction accuracy of the motion energy model. And you can see that there is a separation of voxels. Like, we have some voxels that are really not predicted by the semantic WordNet model, but are very well predicted by the motion energy model. And similarly, we have that some voxels are not predicted by the motion energy model, but they're very well predicted, or they're well predicted by the semantic WordNet model. But then interestingly, we also have some voxels that are equally, also in relative sense, relative to the absolute prediction accuracy within each feature space, but they are well predicted by both feature spaces. And we can see that with a 2D flat map. where basically here we are again on a flat map, but we're coloring each voxel depending on what feature space gave the best prediction in terms of, in this case, R square, coefficient of determination. So you can see that in blue, we have areas in early visual cortex that are better predicted by the motion energy, model. And in orange, instead, we have areas that are more anterior to early visual cortex, extrastriate visual areas, that are better predicted by the semantic model. Now, you can also appreciate that there are some areas that are colored in white, and those are areas that, as we've seen on the 2D histogram, that have high prediction accuracy for both feature spaces. And here, unfortunately, because we fitted two separate models, we have two separate, at least two separate interpretations. One interpretation could be that both feature spaces correspond to different information in the stimulus because they were built in that way. And so their prediction, when we consider it jointly, actually improves the prediction on the brain activity. So this would be the best case scenario where the two features are actually telling us something different about the brain, and together they are better modeling our brain activity. Unfortunately, because we have fitted the models separately, there is another interpretation, which is that maybe because of stimulus correlations, what is being predicted by one feature space is not independent from what is being predicted by the other feature space. And you can imagine a thought experiment where, or actually, where if you were to run an experiment where faces always appear in the same location in your visual stimulus, all your faces are always, let's say, in the top right corner of your visual field, then now you have that the presence or the category face in your stimulus is perfectly collinear with a certain spatial location and perhaps even a certain spatial frequency. In that case, if you consider responses in face processing areas, the motion energy feature space could be actually predicting that responses very well simply because of stimulus correlation, simply because your faces always occur in the same location of the visual field. Now, of course, this is something that you want to avoid when you design your experiment, especially if you use naturalistic stimuli. It's something that you can check. But we have ways to solve this problem or to account for these stimulus correlations using a different type of models. They're not called ridge regression, they're called banded ridge regression. And this represents really an incredible way of, or a very good way of fitting models and to account for multiple feature spaces and for correlations between feature spaces simultaneously. And Tom wrote this package, Himalaya, to support also a paper that's coming up that will actually look at this type of model fitting, which is called multiple kernel ridge regression. The idea of banded ridge regression was already also described in one of the papers from 2019 from our lab by Anwar Nunez Elizalde. And you can look at that paper for more details about banded ridge regression. But here in this last tutorial that I will probably cover in 10 minutes, and I'm probably just going to show pretty figures, I'm actually going to show you how you can use Himalaya to fit these banded ridge models very quickly, actually. And with computation that takes, in general, if you have models with, let's say, 2 or 3 feature spaces, they can be fit in 4 to 6 hours on a GPU. And by comparison, when we were fitting these models before Himalaya on CPU, they could take 1 or 2 days. So it's really very good to use Himalaya, especially if you want to prototype and test different models. So in this last tutorial, we're going to be fitting a banded ridge model with both features, coordinate and motion energy. And again, if you run it on your own on Colab, this is a more intensive model fitting that takes about 6 minutes on GPU. But if you were to run it on CPU, it will definitely last 10 times more. So again, this is one of the cases where if you have access to GPUs, that's a plus for this type of models. So here again, loading the data, everything is the same. For the sake of time and memory, we are computing explainable variance and selecting arbitrarily only voxels that have high EV in terms of like EV higher than 0.1, explainable variance higher than 0.1. In general, of course, we don't do this type of filtering or this type of masking. This is only for the sake of the tutorial. Again, we divide, we mean test set to get an average test set. We make sure the targets are centered. And now this is a difference with respect to the previous tutorials. Here, we're loading both feature spaces at the same time. And I'm going to spend a little bit of time describing how we do this or why we do this, because it can seem complicated, but once you know why we have to do this, it's actually fairly simple. So all scikit-learn models require basically a y, which is our data they want to predict, and an x, which is what we try to predict from. This x needs to be always like a 2D, except maybe there are some exceptions, but in general for these linear models, they need to be two-dimensional tensors or NumPy arrays. So in order to make it, banded ridge work within the scikit-learn API, we basically need to stack our feature spaces, our different feature spaces, into a single matrix of we have as many rows as the number of samples that we have, and as many columns as the number of features. And so here we're basically creating a list that contains XS train, contains all the features in the training set, both WordNet and motion energy, and XS test contains the same features for the test set. And then we're also keeping track of how long or how large they are, so the dimensionality of these feature spaces, because we're going to use this information later to basically tell the pipeline that, hey, that's where we need to split our input matrix to delay the features and create individual kernels that will be passed to the model. So as you can see here, basically our training set, our feature spaces in the training set have 3,600 samples, which is the number of TRs that we have in the training set. But now the number of features is 8,260, which corresponds to the two feature spaces summed together. Again, we define cross-validation scheme. This doesn't change, same as before. But now we're ready to define the model. And here it looks slightly more complicated, but it actually uses the same concepts. The only thing that we're doing is that we're defining a single preprocessing pipeline that then is applied to each feature space. So what we're doing, we're passing this matrix X with both feature spaces. We're splitting it into the two individual feature spaces. We're applying the same preprocessing pipeline. We're computing the kernels out of these feature spaces. And then we're passing the kernels to a Himalaya method called multiple kernel rich CV that will basically optimize the regularization parameters independently for each feature space. One other important thing to know here is that Himalaya implements two different types of solvers to solve this problem. One is a random search, and another one is gradient descent. In general, when we have a lot of targets, like in fMRI, hyperparameter random search is more efficient. So in this case, we're going to be using random search. The problem is that there's no stopping criterion. I'm going to skip this part for a moment. But the important thing about the random search is that we need to define a fixed number of iterations that will be run so that the space of possible combinations of hyperparameters are sampled enough. And because this is random search, let's say we have, in the case where we have two different feature spaces, we're basically sampling pairs of hyperparameters, testing the model with these two hyperparameters, and then repeating it with another random sample. And so there is no stopping criterion, and we have to specify a number of iterations before running the model. Now here, just because it's an example, a tutorial, we use 20 iterations. In general, this is too low for for analysis or models that you want to use in your publications. It's hard to say what a good number is. Again, this is something that you want to test by yourself. In general, I can say that with 2 feature spaces or 3, we definitely use 1 order of magnitude more of iterations. So in the order of 400 to 600. But again, this is something that needs to be tested. And especially if you have more feature spaces, then you want to increase the number of iterations. Another important set of parameters that I just want to briefly mention are these parameters, batch parameters, that basically split our data set in chunks and push the smaller chunks to the GPU so that the GPU memory can actually hold all these data sets. Again, these parameters are something you want to play with because it's very easy to get memory errors when you're trying to load data sets that are too long, they are too big. You really, really want to test this before, let's say, running your model overnight, because otherwise you'll be very sad in the morning when your model is crashed because you hit memory error, and this has happened. So again, really try and see if you hit memory error. Here, for this particular case with Colab, these values are good enough to avoid memory errors. And then again, we define everything as before. The only difference here is that we're using, instead of kernel-rich CV, we're using multiple kernel-rich CV. We need a bunch of extra parameters. There is documentation that you can read. I'm just going to go quickly since we are getting close to the end. And what you end up with at the end, I'm just going to skip to this pipeline. This is our final pipeline that implements banded ridge regression, or if you want, multiple kernel ridge CV, where we're using this, it's called a column transformer, or a transformer, which is called column kernelizer, which what it does is what I said earlier. It basically takes our input features, splits them into 2, and you can actually see, if you click here, you see that they have a parameter that says where exactly to slice our features. So for example, we know that the Warnet features correspond to the first 1700 features, whereas the motion energy to the last 7,000 or so. And then the same preprocessing is applied to both features, and then they're both passed to multiple kernel ridge CV. We can fit our model. Again, if you do this on your own, this is going to take about 6 minutes or more, because we are trying to get random sampling and sample the optimal hyperparameters. In this tutorial, then we're also comparing this multiple kernel ridge CV with a standard ridge model, where we don't do any splitting. We just use one single regularization parameter for both feature spaces. And then we can compare whether we actually get better prediction accuracy, because we went through all these changes. Let's see if we actually get better prediction accuracy. So here in this last couple of cells, we are feeding another pipeline, a baseline pipeline, where we only use kernel ridge CV. We use the same delays. And then we compute the scores, again, on the masked set of voxels that have high EV. And here again, we use a 2D histogram to visualize the prediction accuracies. On the x-axis, we have kernel ridge CV. On the y-axis, we have multiple kernel ridge CV, or if you want, ridge regression and banded ridge regression. And as you can see, banded ridge regression in this case really outperforms single ridge or ridge regression with a single hyperparameter, with a single regularization parameter. Now we can go, banded ridge regression gives us better prediction accuracy, but that's not the only thing that it does, because it optimizes the regularization parameter for each feature space separately. It also allows us to look at the predictions of each individual feature space separately. I'm not going to go into the details, but intuitively, we can think that the prediction y hat of our banded ridge model can be split into the prediction of the individual feature spaces. In this case, we have two feature spaces. So we would have a y hat predicted by the motion energy features and a y hat predicted by the motion energy features. And because these are just linear models, the final prediction is simply the sum of these two predictions. With this understanding, then we can recompute or rewrite the prediction accuracy, the R square, into split R square, which basically it's the amount of variance uniquely explained by one feature space, or saying this in a different way, it's the amount of variance explained by one feature space while accounting for the correlation between the two feature spaces. And mathematically, this allows us to then rewrite the total R square as the sum of the individual split R squares. And because we have this nice decomposition into these quantities, we can then compare the two models. And we can have a better way of comparing the model prediction accuracy of the two different feature spaces. And this is the last flat map that I'm going to show you. Here, we're looking again at the model prediction accuracy of the motion energy model and the WorNet model, motion energy in blue and WorNet in orange. And if you remember, when we plotted the similar flat map using only individual ridge models on the individual feature spaces, there was a lot of white voxels. Now they're most of them have disappeared. And we can see that the motion energy is predicting very well in early visual areas as expected, whereas the semantic model, the WorNet model, is predicting very well in visual semantic areas. And again, this is fit on a reduced data set. So when you will use all voxels, you will appreciate even more differences. And again, all of these particular results are described in the paper, in this 2019 paper. And there are more detailed analysis on what exactly banded ridge regression is doing. But as you've seen, it's fairly easy to fit these models. And yeah, I think we all hope that you can fit these models on your data. And I think this is the end of the tutorial. I don't know if anybody wants to add anything else. I'm going to stop sharing.
Right on time.
Strange for an Italian.
I will applaud on behalf of all of the audience as well.
Perhaps I can add for the recording that at least a few of the organizers have expressed a willingness to continue answering questions about the tutorial. We know it went by pretty fast. So if you have questions on HackMD, you can put those questions on HackMD on the notebook or the pad that's linked there, or potentially on the GitHub repository for the tutorials that Tom has shared, Matteo and Tom have shared.
Well, that's fantastic. Exactly, right on time. And thank you all. This was extremely informative. I feel like I learned 10 times more than I even expected, and I expected to learn a lot. So thank you so much for putting together such a clear and informative and exciting keynote. And the Google Colab was fabulous. I followed along, everything worked great. And I was chatting on the side with some of my students and saying, we should do this. This is very exciting. And now we have all the tools to do it. So thank you again for sharing this with us, with the CCN community, and with anybody who's then watching this recording later as well. This was really fantastic. So do you have any last words you'd like to share before we close up for the day?
Well, I thank everyone actually for participating, for the organizers again, for Matteo and Tom and Mark and Jack, obviously, to be here today and putting all this together.
Yeah, I really appreciate the CCN organizers, Kendrick and Megan, everybody else to allow us to do this tutorial. And I want to thank Fatma and Mark and Matteo and Tom for doing such a great job organizing the really a lot of work on organizing these notebooks. And if any of you try to use these notebooks after the tutorial's over and you have any questions or comments or suggestions, we're finalizing these for publication and we would really appreciate input. So please feel free to get in touch with the developers and let us know what you think. Thanks very much.
Thanks all. Well, this is a fantastic resource. Thank you again for your time, for your energy. I can also agree. I can see how much effort went into this. So thank you for putting it together and sharing with us. And I guess that's it. So we will close up, but I'll remind all of our viewers and anyone watching the recording also that there are 2 more of these Keynote tutorials for CCN this year, and we hope to continue this format also in future years. Don't forget also to come to the generative adversarial collaboration kickoff workshops and the news and progress report this coming Thursday as well. And I think that's all that I need to say. So thanks again, everyone, for being here. Thank you for doing this with us, and we'll see you at the future CCN events over the coming weeks. Take care. Bye.
Thank you. Bye.